Once again, I'm Christine Vokey, Director of Education and Information at PXE International, and I'd like to welcome you to the webinar by Patrick Terry, PXE Research Updated. I'd like to ask everyone to mute their own phones using *6. At the end, when you can ask questions, you can unmute your phone with the *6 command also. I also want to draw your attention to the control panel on the right side of your screen. There is a pane or a box that says questions, and you can ask questions, written questions, by writing them in that box and clicking send. And they would be questions that I can then ask the presenter during the presentation. If you're having any technical difficulties, you can also use the question box to ask me about that, and I'll do my best to help you. And with that, I'd like to introduce our presenter. We are delighted to have Patrick Terry presenting on an update on PXE Research. Patrick is co-founder with Sharon Terry of PXE International. He's also the dad of Ian and Elizabeth Terry. He's very highly qualified and knows all about the research, and he can tell you more about where he's coming from. And Patrick, over to you.
All right. Thanks, Chris. Well, thanks everybody for joining us. What I'll do is I'll race through some slides and talk about PXE Update and some ground rules here, or some outline. What I've done here is I tried to make sure that each slide was numbered. For audio purposes, this is going to be archived on the website, but the slides and the audio will be delinked. So for anyone to follow along, they're going to need to know what slide we're on. So I'm going to try to focus on trying to remember to say what slide I'm on. I'm going to talk about 40 minutes or so. And as Chris has mentioned, there's a box that you could type in your questions, and we can ask as I go. Or if you have any concerns or any other things, comments, just use that question box, and Chris can read those out. And we'll have some time at the end. One big caveat is that this is not medical advice, and I'm not a doctor. I'm just a dad. So some of your questions may be highly specific that I will not be able to answer, but to the extent that I can offer an answer to any of your questions, I will certainly try to do so. At the end of the presentation, there's going to be more information at the end. Chris will get back on the line and discuss things. I'm on a tight schedule that I couldn't rearrange, so we'll try to keep this within the commitments of the time that we've outlined here. So in the presentation itself, what I'd like to just review is I'll go over some background or an overview. I definitely want to talk about basic genetics. Genetics for PXE International have been extremely important, kind of the foundation of why the foundation started and how we've conducted the research enterprise for these many years. Then touch on the genetics of PXE and why that's important, and update you on some exciting research that's taking place with animals and creating animal models of disease for pseudoxanthoma elasticum. And then get right to the substance of this update and talk about the next steps and what PXE International is doing with our clinical and scientific collaborators on interventions and on developing diagnostics and therapeutic approaches for PXE, because ultimately we want to remain focused on what's important in helping individuals that have signs and symptoms of PXE, but also working toward a day that once an individual is diagnosed with early symptoms, that the disease process can be halted or alleviated in some degree, so the most severe outcomes can be avoided. And then I'll also update you about all of this research. While it's very exciting and interesting, does it matter to people and to humans at the end of the day, and talk about the clinical studies that we want to launch this year, and talk a little bit about that. So that's kind of a complete overview of what I'm going to say. But why I'm here and why I'm even talking to you today is because of Elizabeth and Ian Terry were diagnosed at the same day with pseudoxanthoma elasticum with the skin findings, and that was both traumatic and life-altering for my children as well as Sharon and myself, because the way we decided to respond to that diagnosis and what we've done to kind of cope with that diagnosis, both Sharon and I as individuals and as parents and now as organizational leaders, for pseudoxanthoma elasticum and PXE International. So I'm transitioning from slide 4 to slide 5 just to highlight, and for full disclosure, all of my other affiliations, since I am active in the healthcare space in a variety of scenarios, and Sharon, my wife, is as well. Sharon is president and CEO of the Genetic Alliance, which is a Washington, D.C. based umbrella organization that's working on behalf of thousands of disease organizations to try to accomplish the same thing that PXE International is trying to accomplish. Another organization that Sharon and I have founded is called the Genetic Alliance Biobank. So part of what I'll share with you about the PXE story today, we took some of what has worked for PXE and helped bring it to industrial or commercial grade services for other disease organizations. So we created a not-for-profit organization to organize and to do research like PXE International has done with a helping hand. So rather than Sharon and I helping disease groups one at a time, like we used to do for many years, we've decided to kind of build an infrastructure to help them in a much greater degree. I recently have resigned and left the International Genetic Alliance, which is a not-profit organization that was supported by France, the nation of France, and underwritten by the World Health Organization to kind of take patient advocacy, or take the American model or expression of disease-specific patient advocacy, and translate that into national health systems and into emerging nations around the world to help disease groups and patient population organize themselves around the world. And then the last two I'll just briefly discuss. They're the Personalized Medicine Coalition, so this whole aspect of all of this genetic and genomic technology is going to allow for personalization of medicine, and there's lots of challenges there for trying to do this and deliver it to clinical care. So the PMC and the 21st Century Medicine are two additional not-for-profit organizations that I established to kind of help this process along. And the most recent manifestation of what I do is because of the PXE experience, I've learned a lot about research and technology to help my children, and I've taken that knowledge and that new learning, and I started a company a few years ago that's been highly successful that I recently resigned from, because ultimately I'm not really about starting companies, but I'm really focused on wanting just to help PXE and help my children and kind of move on with life. But this experience with genomic health has taught me a lot, and I'm actually in Denver today talking to another company that wants to kind of do the same thing that genomic health has done, so I'm just helping them do the same thing. So that's kind of my side jobs. PXE International and patient-managed research. So the PXE International is a not-for-profit. It's a disease support group, and it's a research organization. So on slide 7 and now slide 8, so PXE International is fundamentally and quite simply a structure to bring patients or participants in the research community together in a meaningful way. And we've decided to use this firewall of separating patients and researchers in a rational way to protect patients' privacy and their family circumstances, as well as to help the research community to do efficient and well-executed and high-quality research as effectively as possible. So basically, PXE International is just fundamentally a facilitator. We bring the lived experience with pseudoxanthoma elasticum to a research and clinical community and ask for help and ask for their assistance to solve and answer questions. So that's basically what the day-to-day activities are for PXE International. On slide 9, one of the aspects of the success for PXE International was the creation of a biobank. So the foundation, after it founded, quickly started a biorepository, a patient registry, started collecting PXE patient samples and medical records, and starting patient organizations around the world to organize PXE across the globe because it's a rare disease. So we felt that if we could organize each and every PXE patient around the world, then it would no longer be a rare disease. It would be a very unique collection of patients and samples with a clinical and medical need that needs answers and support. So the biobank had a significant aspect in that success. On slide 10, I created what's called a logic map, or a research program for PXE International. The year we founded the organization, we created this map, and we decided to do simultaneous research in a variety of these squares. And I won't go into each and every square, but suffice it to say that each of these squares kind of represent either a discipline of science or a technology or a methodology to answer specific questions about PXE, and they're all interrelated and they're all important. To some degree, they're sequential. You can't get to one box without first going through the preceding box, but there's a variety of ways to go to kind of navigate this plan or this map and go from one direction to another, or help inform one activity with another. So we felt, rather than doing sequential step-by-step approach to solving the disease of pseudoxanthoma elasticum, that we would do this integrated or systems approach and see if we could find serendipity or make a phenomenal discovery that would help patients in the near term as well as in the long term. So that's really kind of all of these activities within the first 4 or 5 years. Most of these boxes had activities in them with researchers and clinicians working on developing evidence and other things. And if you have any questions about this, please put them in the box and ask if there's anything confusing here. But basically, this is not the complete answer for PXE, but it's about building our methods and our process to this ultimate destination of a therapeutic intervention. PXE International has been explicit about we're not trying to pursue a cure for PXE, but we want to find interventions. We want to stall the progression of the disease, prevent the severe outcomes that's happened for some patients. But that's really our goal and objective. And really what we're about is being successful in that last box of finding a solution that works for all the patients with pseudoxanthoma elasticum and then kind of turn the lights out. We're not really about supporting an advocacy group or doing this for the rest of our lives. We just want to come to a conclusion here, and some of this research and some of this update is exciting in that regard because we're getting closer and closer to some real compelling answers about interventions and prevention and so forth of PXE. So PXE, simply it's around the world. There's patients all over the world across all kinds of ethnicities and demographics that have pseudoxanthoma elasticum, and it's because when I explain some of the genetics of why that is, it's kind of universal that there is PXE around the world. The biobanks of the organizational structure that PXE International instituted is around the world. We collaborate closely with these various nations and the patient community and the researchers. We wanted to avoid bringing everything back to the United States and saying everything has to be done here by U.S. scientists and by PXE International. We wanted to be much more inclusive and help researchers around the world come to these important questions and answers and not necessarily try to do it ourselves. So we have support offices, patient support offices around the world that vary in their activity and what they do, as well as the research community around the world. On slide 13 is just a quick outline of the number of universities, the types of studies, the numbers of investigators that are working on PXE. This is kind of a time capture a couple of years ago of what was happening with PXE research and the number of industry groups that were involved with various aspects of understanding and getting us to a therapeutic or a treatment for PXE and understanding the genetics of PXE. And this has probably been one of the highlights, at least, of my direct experience with this foundation, is to work with all of these unique people. And the only reason why we're interacting with them is just by happenstance of that they're interested in this disease or they have a family member with the disease, and we've met and wonderful people around the world, and it's been one of the blessings. And Sharon and I joke that all of our best friends have one disease or another. It's just kind of the circle in which we travel in now, but it's been kind of a wonderful experience to understand what is health and what is disease and what does it mean to be a human being and help others through pain and suffering. So it's been a journey. But one of the things that we also focuses on is delivering services and value to the clinical and research community. So we do that by providing funds for research, supporting groups on getting publications, providing technical equipment and computers and technology, and supporting staff and research and junior investigators around the world in the academic setting. So this has been how we kind of induce particular universities or investigators to work on PXE, as we kind of help them along the way by giving them support to work on PXE. And this has been kind of the fundamental involvement of how does a lay patient organization, how does just a dad, work with research investigators and do science, and this is kind of how we do it. We're sometimes 2 or 3 degrees removed from the science, but we're involved with providing these services or these benefits to some of the researchers. At this point, I'll kind of stall a little bit and see if there's any questions. And I wanted to touch on and get into genetics, basically, and then the genetics of PXE. And Chris, if there's no questions, then I'll just proceed.
Sorry, Pat. Yes, there's no questions at this point, so please proceed.
Okay, thank you. So genetics, and why we focused on genetics is, with both our kids diagnosed on slide 16, we quickly realized that it's genetic, that somehow both my kids inherited, altered copies of a gene that causes pseudoxanthoma elasticum. And about that time, the human genome project was underway, the international effort to kind of sequence or understand all of the genetic code that makes life on this planet, and specifically what humans have in each of their cells, their chromosomes and their DNA. And so our human genome is broken up into these little fragments called chromosomes, and we have 2 copies of each of these chromosomes, and the chromosomes are numbered from 1 to 22 by size, with 1 being the largest of the chromosomes and 22 being the smallest. And basically, we have these structures, and 2 of these structures, because we inherit one of these structures from our mother and the other structure from our father. And this is how we pass on genetic code to our offspring and our children. And there's also sex chromosomes, the X and Y chromosomes. So this is an example of a male genome with the X and Y. And the research has established that the PXE gene is located on chromosome 16. And we won't get into the important aspect of how we got there or the time-consuming. Many years, many people worked on going through this entire human genome of these 6 billion letters that makes up our genetic code to try to find which of these genes was important to PXE and why. So that was a big discovery in 1999 and in 2000, and it embarked an effort to understand how genetics is inherited for PXE. And slide 18 just again shows that there's a sperm and an egg, and when they combine, they create this compound genome that makes up of each and every individual. And on slide 19, it outlines, so the genetic code is described about the molecules and the molecular descriptors of the genome are described as A, T, C, and G. These are the letters that make up the spelling of our entire genome. And this spelling, sometimes if you have a misspelling or if you have a G instead of a T in a certain location, sometimes that is what's called an alteration or a mutation. And that misspelling sometimes produces deleterious outcomes or disease outcomes like PXE. So this was an aspect of what PXE wanted to focus on, is the genetics and genomics of what gene causes PXE, and then ultimately how is that gene spelled or misspelled. And you can have different types of misspellings. Sometimes inheritance for genetic disease is described in 2 manners. It's described as a dominant mutation. So if one of your copies of a chromosome or a gene has an error in it, if it's a dominant mutation or an alteration, then an individual will have disease. But in most diseases, the inheritance is called recessive. And recessive means, quite simply, that you potentially have a good copy and an altered copy of a particular gene. And in that case, if it's a recessive disease, the good copy can compensate for the loss or the alteration in the bad copy, and you don't have disease. You're just what's called a carrier. The carrier means that you have this recessive mutation in your genome, and you're carrying it, and it doesn't really matter that you're carrying it because you've got a good copy and all is well with the world. But in the circumstance like my children, in a recessive disease, what happens is both our children inherited an altered copy from Sharon and an altered copy from me. So both my children have two errors in the PXE gene in their genome, and that's how they have PXE. So both Sharon and I are carriers. So I have a particular alteration in one of my copies, and Sharon has a different alteration in one of her copies. And as luck would have it, our two children inherited both altered copies and have disease. So that's how we can explain how, Sharon doesn't have PXE and I don't have PXE, and Sharon's family doesn't have PXE and my family doesn't have any history of PXE, and how is it that my children can have PXE. And here's an example of everyone, of a carrier delivering an individual and gets disease at the end when they inherit these copies, two altered copies. So that's kind of a long-winded way of just describing how PXE has been understood. Originally, there was a significant amount of controversy about dominant PXE, but we've shown through science and clinical studies that there is no such thing as dominant PXE, that there is recessive disease only with PXE. Slide 21, just to reiterate that PXE is a recessive disease. And you can still sometimes see in the medical literature someone discuss or report a dominant case of PXE, but it's not the case. It's just basically confusion. But the way in which PXE is transferred from one generation to the next is through this recessive mechanism. Slide 22, kind of moving on to the PXE gene find in 1999 and 2000 and what that meant. There was decades worth of work and lots of groups that worked on understanding and trying to find the PXE gene and trying to understand it. And there's lots of publications along the way here and lots of very interesting people from around the world that worked on finding the gene and reporting it and doing that work in isolating it to a certain gene, which I'll talk a little bit more about what is this gene. So this is slide 24 is a little cartoon of a cell. So if you're going to take a cell and chop it in half, this big blue plasma membrane, as it's called, is kind of the skin or the outside of the cell, the membrane of the cell. And the PXE gene codes for a protein that the gene is expressed in the nucleus, and then it's transported into the cytoplasm or the interior of the cell, and then that gene produces a protein that then locates itself in the plasma membrane or creates this channel protein called MRP6 or ABCC6. So that's the descriptor, the kind of the scientific descriptor of the PXE gene is ABCC6. And we talked about complex inheritance. Sorry, I meant to eliminate that slide. But slide 26. So what is ABCC6, and what does ABC mean? If this is a transport protein, it's very technically called an ATP binding cassette transporter. That's what the ABC stands for. And the A and the B and the C stand for ATP binding. So ATP is a compound that is like a fuel source for the cellular machinery inside the cell. It's literally like a gasoline or a pump engine, and it makes the protein function. And it belongs to a very large family of proteins that have been conserved. So evolution has used these proteins over the eons for lots of different things, from the simplest singular cellular animals to bacteria to plants, and now, as we know it in humans, these proteins are extremely important for a variety of functions. And these transmembrane or these transport proteins function for a variety of reasons, and they are important to metabolism. They're important for transporting products from inside of cells and between cells and in between tissues and in between organ systems. So they have a variety of functions, including, how we respond to different drugs, whether that's, Tylenol or aspirin. These proteins are extremely important for normal functioning biology, as well as all sorts of different human diseases like cystic fibrosis and multi-drug resistance, and of course PXE. On slide 27 is another cartoon of that same protein, but if you took the protein and kind of unfolded it and laid it flat out on a piece of paper, you have this complex structure of this ABC6 protein. And why this is important, because to look at genes and proteins in this fashion help to understand how the gene functions, where it functions, and what happens when it's not functioning, or if it's functioning in a partial way, what's actually happening, what's going on. Because the more you understand how something operates, the more you can understand how you could correct it or enhance how it functions. And we also understand this, this mechanism of this ATP binding of making this protein work. And I think I have another cartoon here on the next slide. No, I don't. It must be further down here. I have a cartoon that kind of shows really what this protein does. But on slide 28, one of the real phenomenal things that PXE International was able to do in collaboration with industry is we were, through the biobank and through our clinical collaborators, PXE International purchased 5 of these highly sophisticated technology platforms and sent them around the world so we could do PXE research around the world on the same technology, with the same methods, with the same reagents and processes to help everyone understand what's happening with this gene and this protein in the same fashion, in the same manner, at the same time. So we wanted to have be free from competition and really be about what matters and focusing on the science and what matters to patients rather than competing with different research groups. And that's certainly one of the highlights of recent experience with PXE International. That was a groundbreaking endeavor, and hundreds of thousands of dollars were expended and donated by this transgenoma company to make that happen. So that was an exciting initiative that has bared fruit. And that technology allowed us to take a look at this protein. Here it is again, this cartoon of this protein. And all these little arrows and numbers are examples of errors in the protein or mutations. So these different things show up with different PXE patients, have different mutations all along this protein and affect the protein in different ways. That kind of explains some of why PXE is expressed differently between PXE patients, because sometimes patients have different mutations and different alterations, and so different consequences to their genetics and to the protein and its function, depending on where these alterations happen. So there's now hundreds of alterations in the gene that have been described and have been studied, and we're working in an ongoing fashion with a number of groups to understand the consequences of these what are called genotypes. They're basically the specific errors that happen for a particular patient. Here's a little out of order, but here's that slide 13 of this is literally what the PXE protein does. It's a pumping mechanism. It's called what's an efflux pump. An efflux just means that it's an exporter. It's discharging something from inside the cell to outside the cell with this two-pump mechanism. And this is how the protein kind of opens and closes and facilitates a transport. And the big question is, at least in this cartoon, what are those yellow dots? And what do those yellow dots mean? And what's being transported and why? And if this protein is not functioning or those yellow dots aren't transported, then what happens? And how does that produce what we know as PXE and diagnose as PXE? So that's been the ongoing activity for the past 3 years with the research community, is to try to understand what that is, and I hope to give you some updates on this. One of the other things here, this is what's called a family tree, or a phylogenetic tree, and there's 48 different proteins in the same family. So through evolution, all of these ABC transporters have diversified and developed unique functions, and you can see off in the left that box on slide 31 with the ABCC6. So there's the PXE protein, and there's lots of cousins that it has. And with the research establishment now, PXE International has been involved with studying 16 of these other proteins to try to understand, is there a way to enhance or upregulate one of these functioning proteins with PXE patients to compensate for loss of ABCC6, for instance. So that's one of the research initiatives, to see if there's a way we can compensate for the loss of function for this protein. And that's been very interesting because there's lots of other human diseases involved here. So on slide 32, if you look at all these different transporters here on the right, all these different numbers represent different transporters and genes, and then on the left, there's different diseases that are associated with these proteins: cancer and age-related macular degeneration and Stargardt's disease and cystic fibrosis. So this whole list of different diseases, including PXE, are associated with these proteins. And this has been a very interesting experience because over the last 5 years, most of all of the transport protein science and researchers have been studying basic science and have not been involved with human research studies or looking at patients. And part of the PXE International work has been to bridge that gap, is to bring all of these basic scientists that are working on these transporters here. So I can highlight all of these transporters. If you think about, there's thousands of scientists working on all these transporters that have never seen a patient with disease of any sort. And so part of the education process is to say, well, you're working on ABCC6 in yeast or in mice or in basic research settings, and we want to introduce you to PXE International, we want to introduce you to PXE, and here's our condition, here's our circumstance, here's our problem, would you be willing to work on PXE and help us? So that's been gratifying because a lot of the basic science and research community has been very willing to support and get involved and contribute their time and energy to help PXE. So all of this genetics, part of the research endeavor on slide 33 was to see if we could, our hope was that we could link a certain genetic alteration in the PXE gene to the type of PXE that an individual patient would have, or at least be able to describe to a patient how severe their disease might be, what's the probability of certain clinical complications, or are there other correlations we could help make and inform doctors and patients about PXE. But we worked really hard with a whole bunch of collaborators, including the federal government in the United States and the Center of Disease Control, to look at the genetics of PXE and all of these mutations that we described and the disease manifestations, and see if there's what's called a genotype-phenotype correlation. And phenotype just means kind of signs and symptoms in a human being. What does PXE look like? And do the mutations in the gene correlate to the signs and symptoms? But very disappointingly, we've discovered that biology and the disease of PXE are much more complex than that, and we can't answer those questions directly. But there are some subtle questions that we can ask and answer around the genetics of PXE and certain decisions around how PXE can be managed in the future. On slide 34, so the results so far since 1995, when the research endeavor started for PXE Foundation, we worked hard with the gene discovery. We created these databases for research. We created the biorepository of patient samples and the ongoing patient registry. We launched this aggressive initiative about the genotype-phenotype program. We created this firewall and this clinical structure to help protect patients and their privacy. We worked with institutional review boards and institutions, with participant protections, and then we worked aggressively around kind of the business of science and about patenting and licensing. So the PXE Foundation went about and patented the PXE gene and licensed it to industry to help facilitate research on PXE as opposed to hoarding it or taking a monopoly. So we actually use patenting to help advance research as opposed to preventing research from occurring. We have the clinical diagnostic program with an industry collaborator for clinical diagnostics for PXE. I'll share with you a little bit more in the slides here about the animal studies, about what's called a functional assay or the drug discovery program, and then finally quickly touch on what we hope to bring into fruition in the next couple of years as the human clinical studies and drug and treatment experimentation in humans for PXE. So the research, we're studying humans and animals with PXE on slide 36. And sometimes one of the frustrating things is when you create an animal model for a certain human disease, sometimes it doesn't behave the same way that the human disease behaves, that it manifests differently. For whatever reason, mice and other animals are just different from humans, so it's not all that informative. But luckily, in the PXE experience, the animal models are quite in line and in parallel with the human disease, which is an exciting development. So on slide 37, there are actual research groups that have PXE mice and have PXE zebrafish. And zebrafish are really interesting. They're these tiny little minnow-like fish that are, their embryos are clear. Their young adults are clear, so you can actually see through them. So you can see how PXE manifests itself and how the zebrafish will, some of its organs and systems will mineralize and calcify just like human aging does, but you can create these, there's labs now that have hundreds and hundreds of tanks with thousands and thousands of zebrafish, and they're doing a variety of studies with mice and with zebrafish for different types of approaches with manipulation, with all kinds of different drug studies to see if there's an actual way where you can cure or eliminate Pseudoxanthoma elasticum in these animal models and then hopefully translate that into human intervention. So that's in the mouse itself is about 3 years old now. The zebrafish was just established the end of 2008. So all of these models are very new. There's the mice models around the world now. PXE International helped facilitate distribution of these mice through different research groups around the world. So there's a whole number of research groups working with these mice and trying to understand what does PXE do to mice and how they can reverse it. One of the discoveries has also been that this transport protein operates on slide 38 here in the liver, and the function of this protein, or when this protein is not functioning in the liver, something is or is not transported into the circulatory system that causes all of the signs and symptoms of PXE. So here is, the circumstance with PXE having mineralization or calcification of what's called the extracellular matrix. So the skin fibers, the vascular lining of the blood vessels, the cardiovascular system, all of the organ systems affected by PXE are impacted because the extracellular matrix is mineralized, and the elastin and the collagen fibers in these tissue systems mineralize. And it's like an advanced aging process. It shouldn't happen, but it does. So understanding how this functions and what is inside the liver that's being transported or not transported, how the liver functions or doesn't function correctly for PXE patients is where the research is focused currently to try to understand that in a rigorous way and to make sense out of it. On slide 39, so it's been described now after the gene was found that it is a metabolic disorder, that most likely this gene is not functioning correctly in the liver, and all of the signs and symptoms of PXE are secondary events to this underlying liver malfunction. So there's lots of research taking place in what's happening in the liver or not happening in the liver in PXE patients, and what does metabolism mean for PXE patients. And it's not about being a slow metabolizer or a fast metabolizer, but it's about how certain molecules or nutrients are actually processed by the body that are different for PXE patients than patients that don't have alterations in this gene. The diagnostic testing I've already talked about a little bit, that it's available and clinicians are using it with clinical collaborators, and we're working to refine and enhance that so physicians can look at an individual and say, well, it looks like you have PXE, but I'm not really sure. Let's do a diagnostic test and find out. Or for a family that has a child with PXE and they want to understand who's a carrier, or if another child may be affected, there are different uses for using the diagnostic test. But in the research setting, it's important for us to use this technology in all of those patients that we screened on that technology platform that I described earlier, is to understand how these alterations in the gene affect what the protein can and can't do, and does and doesn't do in the cell and in the liver. So the diagnostic testing has been important to help us understand kind of the diversity of alterations and how this PXE behaves differently in different patients in the research setting. The animal studies and the intervention approaches are pretty, they're in early stages. It's preliminary work. There's been a couple of publications just at the end of this year about research in this area, so it's brand new. It hasn't been verified and replicated by other groups, so a lot of this information has to be kind of discounted as preliminary or exploratory, and there's much more work and rigorous effort that needs to be applied to understand what is and is not true, and what may work in mice and zebrafish may not translate to humans, those types of things. So there's a lot more work that has to happen in the next few years, but certainly a lot of interest to accelerate this process and get to those answers. And also doing drug discovery. So is there understanding now of what this protein does and doesn't do and where it operates? Are there existing drugs that may work for PXE patients? So there's a whole drug discovery program and research happening with what's called artificial cellular assays. So we make this artificial PXE cell, and you can expose it to all kinds of drugs and therapeutics or conditions and see if you can improve the function of that protein through drug therapy. We did get a question about ahead of the webinar about eye interventions and eye treatments, and that's certainly a significant amount of effort that Sharon has been engaged in with the National Institutes of Health, of creating an eye intervention clinical program at the NIH Clinical Center in Bethesda, Maryland. On slide 44 is just kind of a picture of the giant hospital setting and research institution that Sharon's been working with and trying to put a clinical program together in getting that launched in 2009. On slide 45, these are kind of all of the open questions that are currently active now in the research community and within PXE International, is to understand what's the natural function, what's being transported by the ABCC6 protein, what is the liver function, how is it affecting metabolism, is working closely with European investigators to understand, is there tissue-specific functions for this protein outside of the liver, and does that matter for PXE? To understand the expressivity or expression of the disease in both animals and humans, and comparing and contrasting the two. What's termed extreme or intermediate phenotype. So can we get to a point where we can actually get closer and closer predicting PXE without signs and symptoms? That's important to build that evidence around. Can we get there? I talked a little bit about the genotype phenotype, but that's still ongoing. The protein structure, the modifications, are there other genes that are involved with PXE and that matter to PXE? So this whole idea of what's called gene modifiers. Are there other genes that influence PXE? That work is ongoing. And then an important influence to understand is, does environment matter? And to what extent does it matter for PXE patients? So if you have PXE, or if you're even a carrier and you're a chain smoker, there's an environmental influence or an insult that could exacerbate disease or make you prone to certain complications. So understanding and getting that clinical work done is also important for us. And certainly the human clinical intervention trial that Sharon is working on, that's coming together nicely. And there's also a lot of ongoing work anecdotally happening out there with approved therapeutics that are on the marketplace from a pharmaceutical company called Genentech, with therapies called Avastin and Lucentis for the eye lesions and the eye complications of the bleeding for PXE. And those efforts are happening. There's a lot of anecdotal and rumors about its success and its complications. So one of the aspects that Sharon is working closely with the NIH on is, can we actually produce a rigorous controlled clinical trial to actually show that one of these therapies can work for PXE patients, and if so, how, and how should it be delivered? And once we do that, then the therapy can become approved for that indication, be reimbursed by insurance companies, and be a viable treatment for people with advanced eye disease. And then I won't even go through this litany because it's kind of a repeat of what we just discussed here, is that, you know, all of these are current projects here on slide 46. They're underway now. They're highly active in 2008. They'll be even more active in 2009. There's lots of research monies that we're trying to apply for and get, and we're hounding the PXE community all the time for donations, for allowing us to get this work accomplished. But it's all proceeding. It's happening at a slow pace. It certainly could happen. You know, if money was not an object, it could happen more quickly. But there are certainly research and resource limitations for the foundation that we can only do so much. But it's really exciting what's happening and what's taking place out there. And I think slide 47 may be my last slide. It is. And slide 47 is just looking at, and I'm a little hesitant to talk about this, but I want to share with you that there's, in the mice, there's very interesting evidence about the dynamic interplay with phosphate and magnesium and vitamin K. So these are almost to the extent that you can think about vitamin therapy impacts the severity of the disease in mice, and we need to understand what's the dynamic interplay of the liver function and these different minerals and compounds, and, you know, how does it impact mineralization and kidney function in PXE. And there's certainly a lot of work, and there'll be more and more publications coming out in the next few months about vitamin K and its influence on what the MRP6 gene is doing and not doing with vitamin K metabolism, for instance. So you'll see a lot more information in PXE International newsletters and annual meetings moving forward. So that's the end of the slides, and I'll finish there. And if you have any questions, please ask away.
Pat, I don't have any written questions, so I'm opening questions to the audience. I am unmuting you from my end right now, and if you unmute yourself using *6, please feel free to ask a question.
Yes. Are there any parallels between PXE and cystic fibrosis? I know that the proteins are very different, but I noticed that, you know, it's ABCC6 for PXE, and it's, or vice versa, and ABCC7 on the other.
Yeah, that's a great question.
What are they, if they exist, please?
Yeah, it's a great question. The proteins, while they're similar in their kind of the recipe that makes up these proteins, they are different in their function. So cystic fibrosis gene and protein operate quite differently in the body than the PXE gene, but the the CF Foundation and the CF community is way out ahead, almost by 20 years, in their research and their activities. So a lot of what they've done with drug discovery and protein work is helping PXE International and our researchers. So we're working quite closely with not only the CF Foundation, but the research community that has worked historically on CF are now migrating and starting to work on PXE. So there's lots of overlap. It's not directly related, but it's really important.
Thank you very much. I have a nephew, a grandnephew that has cystic fibrosis, and I have a husband with PXE. So I was very fascinated because I'm interested in both conditions. Thank you so much for your help.
With the PXE, any questions? What is it that he noticed that supposedly these problems start in the liver. So how does it get up to my eyes so that causes me my bleeds?
Right. So that's one of the big questions: is how does an alteration in your liver function cause vision loss or cardiovascular difficulties or skin wrinkling. And that's the challenge here, to understand there is probably something circulating or not circulating in our blood system that is causing all of the complications of PXE. So one of the hypotheses or the theories is that there is, if you think of it quite simply, as there's a circulating microscopic scissors that are circulating through your body that are cutting collagen and elastin and creating this damage to those cellular and tissue systems, and from that damage results PXE. So naturally, any damage to the skin or the body, mineralization happens, calcification happens. It's part of the healing process. But for whatever reason, this damage that's caused by this microscopic scissors doesn't get repaired and becomes permanent, and does damage to the Brooks membrane in the back of the eye, and does damage to the skin and the laminal layer of the blood vessels, and this mineralization causes PXE. So the big question is, how do we get from the signs and symptoms to PXE and explain it to a liver function? So there's certainly a lot of work and a lot of theories and experimentation that's happening to try to answer that question.
Can I make a follow-up question regarding the liver? Just about everything, as you know, Patrick, people who suffer from PXE suffer from the usual line of stuff as well.
Right.
I of course, have got the stent, and I've got the diabetes, and I've got you name it. Okay, I've got it. But it seems like all of and also they want to lower my cholesterol, and all the cholesterol stuff goes into the liver, all right, and works in the liver. All of those medicines. And what I'm concerned about is, are any of these medicines I'm taking, such as Plavix and well, I was on Lipitor and all the 9 yards of those until I couldn't do them anymore, and all of the metformins and all of the other stuff that apparently work in the liver. Are we looking to see whether or not PXE is affected by any either effects or gets affected any of these meds? My own reason for asking that was because when my doctor started playing with the cholesterol medicines and the diabetes medicines, I started to get in a bleed in an eye that I hadn't got a bleed in. But I know that there's no correlation, but that's just anecdotal. But
right.
That's my question.
Well, I can't answer that question as being a non-medical professional to say that there's no complications or no side effects or no problems. But one of the immediate questions came up when the whole idea of vitamin K metabolism is that what the PXE gene is doing, is processing some component of vitamin K or some conjugate of vitamin K. Someone immediately thought of bleeding disorders, and what happens if patients are taking warfarin? Do they have serious adverse events? So the preliminary research in that regard has proven negative. So at least in what you would think would be a complication in PXE hasn't borne out. So there's no evidence that there's unique complications for PXE patients for taking Lipitor or any other med at this point.
Pat, I have a question that's been written in from Gordon. He asks if there are any examples of PXE patients who had a liver transplant either before or after PXE symptoms were noticed.
Yeah, that's a very intriguing question. We have an opposite example of that in real-world conditions in 2 patients where individuals received a liver transplant, and as it turned out, they've received a liver transplant with someone with PXE. So those patients, when they received the PXE liver, developed signs and symptoms of PXE. So they needed a liver transplant for some other reason, but there's a case in France and a case in the United States that demonstrates and then kind of proves the principle that PXE is a liver disease.
Okay, thanks, Pat.
Are there any other questions? Well, I don't have any more written questions, so with that, our webinar has concluded. I want to thank Patrick Terry for updating us today on the research going on in PXE, and thank you all for coming.